Dear students, hello and welcome. Today I am going to deliver a lecture on hematopoiesis, T and B lymphocytes, natural killer cells, monocytes and macrophages. Let's first try to have a look at the main objectives of today's deliberation. To give a general and thorough idea about the process of hematopoiesis, explaining in detail the structure, functions and types of T lymphocytes, to discuss in detail the structure and functions of B lymphocytes, to give a general idea about natural killer cells and monocytes, finally discussing in detail about macrophages and their functions. Now we will start with the brief idea about the process of hematopoiesis. Hematopoiesis is the formation of blood cellular components. All cellular blood components are derived from hematopoietic stem cells. In a healthy adult person, approximately 10⁻11 to 10⁻12 new blood cells are produced daily in order to maintain steady state levels in the peripheral circulation. Hematopoietic stem cells reside in the medulla of the bone, that is bone marrow, and have the unique ability to give rise to all of the different mature blood cell types and tissues. Hematopoietic stem cells are self-renewing cells. When they proliferate, at least some of their daughter cells remain as hematopoietic stem cells, so the pool of stem cells is not depleted. This phenomenon is called asymmetric division. The other daughters of hematopoietic stem cells, that is myeloid and lymphoid progenitor cells, can follow any of the other differentiation pathways that led to the production of one or more specific types of blood cells, but cannot renew themselves. The pool of progenitors is heterogeneous and can be divided into two groups: long-term self-renewing hematopoietic stem cell and only transiently self-renewing hematopoietic stem cell, also called short-terms. This is one of the main vital processes in the body. All blood cells are divided into three lineages. Erythroid cells are the oxygen-carrying red blood cells. Lymphocytes are the cornerstones of the adaptive immune system. Myelocytes, which include granulocytes and megakaryocytes, and macrophages. In developing embryos, blood formation occurs in aggregates of blood cells in the yolk sac called blood islands. As development progresses, blood formation occurs in the spleen, liver and lymph nodes. When bone marrow develops, it eventually assumes the task of forming most of the blood cells for the entire organism. However, maturation, activation and some proliferation of lymphoid cells occurs in the spleen, thymus and lymph nodes. In children, hematopoiesis occurs in the marrow of the long bones such as femur and tibia. In adults, it occurs mainly in the pelvis, cranium, vertebrae and sternum. In some cases, the liver, thymus and spleen may resume their hematopoietic function if necessary. This is called extramedullary hematopoiesis. It may cause these organs to increase in size substantially. During fetal development, since bones and thus the bone marrow develops later, the liver functions as the main hematopoietic organ. Therefore, the liver is enlarged during development. In some vertebrates, hematopoiesis can occur wherever there is loose stroma of connective tissue and slow blood supply, such as the gut, spleen and kidney. Now let's discuss the next part of our today's lecture, that is T lymphocyte. A lymphocyte is one of the subtype of white blood cell in the vertebrate's immune system. Lymphocytes include natural killer cells, T cells and B cells. Lymphocytes can be identified by their large nucleus. A T cell or T lymphocyte is a type of lymphocyte that plays a central role in cell-mediated immunity. T cells can be distinguished from other lymphocytes such as B cells and natural killer cells by the presence of a T cell receptor on cell surface. They are called T cells because they mature in the thymus from thymocytes, although some also mature in the tonsils. There are several subsets of T cells, each have a distinctive function. The majority of human T cells rearrange their alpha and beta chains on the cell surface receptor and are termed as alpha-beta T cells, which are part of adaptive immune system. Specialized gamma delta T cells have invariant T cell receptors with limited diversity that can effectively present antigens to other T cells and are considered to be part of the innate immune system. Now let us discuss their types. Number 1. Effector T cells. The category of effector T cells is a broad one that includes various T cell types that actively respond to a stimulus such as co-stimulation. This include helper, killer, regulatory and potentially other T cell types. Number 2. Helper T cells. T helper cells or Th cells assist other white blood cells in immunological processes including maturation of B cells into plasma cells and memory B cells and activation of cytotoxic T cells and macrophages. These cells are also known as CD4 positive T cells because they express the CD4 glycoprotein on their surfaces. Helper T cells become activated when they are presented with peptide antigens by MHC class II molecules, which are expressed on the surface of antigen presenting cells. Once activated, they divide rapidly and secrete small proteins called cytokines that regulate or assist in the active immune response. Number 3. Cytotoxic or killer T cells. Cytotoxic T cells destroy virus-infected cells and tumor cells and are also implicated in transplant rejection. These cells are also known as CD8 positive T cells since they express the CD8 glycoprotein at their surfaces. These cells recognize their targets by binding to the antigen associated with MHC class I molecules which are present on all the surfaces of nucleated cells. Number 4. Memory T cells. Memory T cells are a subset of antigen-specific T cells that persist long-term after an infection has resolved. They quickly expand to large number of effector T cells upon re-exposure to their cognate antigen, thus providing the immune system with memory against past infections. Number 5. Regulatory or suppressor T cells. Regulatory T cells are crucial for maintenance of immunological tolerance. Their major role is to shut down T cell mediated immunity towards the end of an immune reaction and to suppress autoreactive T cells that escaped the process of negative selection in the thymus. And number 6 is natural killer T cells. Natural killer T cells or NKT cells, not to be confused with natural killer cells of the innate immune system, bridge the adaptive immune system with the innate immune system. Unlike conventional T cells that recognize peptide antigens presented by major histocompatibility complex molecules, NKT cells recognize glycolipid antigen presented by a molecule called CD1D. Once activated, these cells can perform functions ascribed to both Th and Tc cells. They are also able to recognize and eliminate some tumor cells and cells infected with herpes virus. Now let's try to learn about B cells. B cells, also known as B lymphocytes, are a type of white blood cell of the lymphocyte subtype. They function in the humoral immunity component of the adaptive immune system by secreting antibodies. Additionally, B cells present antigen and secrete cytokines. In mammals, B cells mature in the bone marrow, which is at the core of most bones. In birds, B cells mature in the bursa of Fabricius, a lymphoid organ. B cells, unlike the other two classes of lymphocytes, T cells and natural killer cells, express B cell receptors on their cell membrane. B cell receptors allow the B cell to bind to a specific antigen against which it will initiate an antibody response. B cells develop from hematopoietic stem cells that originate from bone marrow. Hematopoietic stem cells first differentiate into multipotent progenitor cells, then common lymphoid progenitor cells. From here, their development into B cells occurs through several stages. B cells undergo two types of selection while developing in the bone marrow to ensure proper development. Positive selection occurs through antigen-independent signaling involving both the pre-B cell receptor and the B cell receptor. If these receptors do not bind to their ligand, B cells do not receive the proper signals and cease to develop. Negative selection occurs through the binding of self-antigen with the B cell receptor. If the B cell receptor can bind strongly to self-antigen, then the B cell undergoes one of the four fates, that is clonal deletion, receptor editing, energy, or ignorance. This negative selection process leads to a state of central tolerance in which the mature B cells do not bind with self-antigens present in the bone marrow. To complete development, immature B cells migrate from bone marrow to the spleen as well as pass through two transitional stages, T1 and T2. Throughout their migration to the spleen and after spleen entry, they are considered as T1 B cells. Within the spleen, T1 B cells transition to T2 B cells. T2 B cells differentiate into either follicular B cells or marginal zone B cells depending on signals received through the B cell receptor and other receptors. Once differentiated, they are now considered mature B cells or naive B cells. Now let us discuss another part of our today's lecture, that is natural killer cells. Natural killer cells are a type of cytotoxic lymphocyte critical to innate immune system. The role natural killer cells play is analogous to that of cytotoxic T cells in the vertebrate adaptive immune response. Natural killer cells provide rapid response to viral infected cells acting at around 3 days after infection and respond to tumor formation. Typically, immune cells detect a major histocompatibility complex presented on infected cell surfaces, triggering cytokine release, causing lysis or apoptosis. However, natural killer cells are unique as they have the ability to recognize stressed cells in the absence of antibodies and MHC, allowing for a much faster immune reaction. They were named natural killers because of the initial notion that they do not require activation to kill cells that are missing self markers of MHC class first. This role is especially important because harmful cells that are missing MHC first markers cannot be detected and destroyed by other immune cells such as T cells. Natural killer cells are defined as large granular lymphocytes and constitute the third kind of cells differentiated from common lymphoid progenitor generating B and T lymphocytes. Natural killer cells are known to differentiate and mature in the bone marrow, lymph nodes, spleen, tonsils, and thymus, where they enter into the circulation. Natural killer cells differ from natural killer T cells phenotypically by origin and by respective effector functions. Often, natural killer T cells activity promotes natural killer cell activity by secreting interferon gamma. In contrast to natural killer T cells do not express T cell antigen receptors, pan-T cell markers, CD3, surface immunoglobulin or B cell receptors, but they usually express the surface markers like CD16 and CD56 in humans. In addition to the knowledge that natural killer cells are effectors of innate immunity, recent research has uncovered information on both activating and inhibitory natural killer cell receptors, which play important roles including self-tolerance and sustaining natural killer cell activity. Natural killer cells also play a role in adaptive immune response. Numerous experiments have worked to demonstrate their ability to readily adjust to the immediate environment and formulate antigen-specific immunological memory fundamental for responding to secondary infections with the same antigen. The role of natural killer cells in both the innate and adaptive immune response is becoming increasingly important in research using natural killer cell activity and potential cancer therapies. Let's now discuss about monocytes. Monocytes are the largest type of leukocytes and can differentiate into macrophages, and they can acquire some features of dendritic cells. As a part of the vertebrate innate immune system, monocytes also influence the process of adaptive immunity. Monocytes are amoeboid in appearance and have a granular cytoplasm. Containing unilobar nuclei, these cells are one of the types of mononuclear leukocytes which shelter azurophil granules. The archetypical geometry of monocyte nucleus is ellipsoidal, metaphorically bean-shaped or kidney-shaped, although the most significant distinction is that the nuclear envelope should not be hyperbolically perforated into lobes. Contrast to this classification occurs in polymorphonuclear leukocytes. Monocytes compose of 2 to 10% of all leukocytes in the human body and serve multiple roles in immune function. Such roles include replenishing resident macrophages under normal condition, migration within approximately 8 to 12 hours in response to inflammation signals from sites of infection in the tissues, and differentiation into macrophages or dendritic cells to effect an immune response. In an adult human, half of the monocytes are stored in the spleen. Monocytes are generally identified in cystine smears by large kidney-shaped or notched nucleus. These change into macrophages after entering into appropriate tissue spaces and can transform into foam cells in endothelium. Monocytes are produced by the bone marrow from precursors called monoblasts that differentiate from hematopoietic stem cells. Monocytes circulate in the bloodstream for about 1 to 3 days and then typically move into tissues throughout the body. Half of them are stored as a reservoir in the spleen in clusters in the red pulp called cords of Billroth. In the tissues, monocytes mature into different types of macrophages at different anatomical locations. Monocytes are the largest corpuscles in the blood. Monocytes which migrate from bloodstream to other tissue will then differentiate into tissue-resident macrophages or dendritic cells. Macrophages are responsible for protecting tissues from foreign substances but are also suspected to be important in the formation of important organs like heart and brain. They are cells that possess a large smooth nucleus, a large area of cytoplasm, and many internal vesicles for processing foreign material. Monocytes and their macrophages and dendritic cell progeny serve 3 main functions in the immune system. These are phagocytosis, antigen presentation, and cytokine production. Now at last, let's try to learn about macrophages. Macrophages are a type of white blood cells that engulf and digest cellular debris, foreign substances, microbes, cancer cells, and anything else that does not have the type of proteins specific of healthy body cells on the surface in a process called phagocytosis. These large phagocytes are found in essentially all tissues, where they patrol for potential pathogens by amoeboid movement. They take various forms with various names throughout the body. For example, histiocytes in connective tissue, Kupffer cells in liver, alveolar macrophages in lungs, microglia in central nervous system, but all are the part of mononuclear phagocyte system. Besides phagocytosis, they play a critical role in non-specific defense, that is innate immunity, and also help initiate specific defense mechanisms, that is adaptive immunity, by recruiting other immune cells such as lymphocytes. For example, they are important as antigen presenters to T cells. In humans, dysfunctional macrophages cause severe disease such as chronic granulomatous disease that result in frequent infections. Beyond increasing inflammation and stimulating the immune system, macrophages also play an important anti-inflammatory role and can decrease immune reactions through the release of cytokines. Macrophages that encourage inflammation are called M1 macrophages, whereas those that decrease the inflammation and encourage tissue repair are called M2 macrophages. Human macrophages are about 21 micrometers in diameter and are produced by the differentiation of monocytes in tissues. When a monocyte enters damaged tissue through the endothelium of a blood vessel, a process known as leukocyte extraction occurs. It undergoes a series of changes to become a macrophage. Monocytes are attracted to a damaged site by chemical substances through chemotaxis, triggered by a range of stimuli including damaged cells, pathogens, and cytokines released by macrophages already at the site. At some sites such as testes, macrophages have been shown to populate the organ through proliferation. Unlike short-lived neutrophils, macrophages survive longer in the body, up to a maximum of several months. Now the functions of the macrophages. Number 1, they have a role in phagocytosis and in adaptive immunity. Number 2, they also have a role in muscle regeneration and wound healing. Number 3, besides they also have a role in limb regeneration and iron homeostasis. That's all about today's lecture. Hope you have understood it well. Thank you very much and goodbye.